time, I'll mention Research Computing Wiki. If you go under Digital Science Education, here we have list of all previous webinars, and they recorded on YouTube. If you want, you can go and select watch for later, or put on your watch list, and you don't have to be on the VPN anymore to see them. All of them recorded here, very nicely organized by Shira. Thank you. So with that, I'll stop my screen share, and I'll pass the mic to Jason Flanick from Boston Children's, who's going to introduce our speaker today. Thank you, Jason, for joining us.
Yeah, sure, no problem. So yeah, it's great to have Dennis Lal here to talk about his work. For those who don't know him, Dennis is an assistant professor at Cleveland Clinic and Case Western Reserve University. It's quite interesting, though. He also has appointments where he's very active at the Broad Institute, and also as a group leader at Cologne Center for Genomics, where he's been for quite some time. And his work focuses on understanding the genetic contribution to brain disorders, applying a lot of different approaches ranging from computational work to molecular biology and clinical research. I'm really interested to see this talk. I've been following Dennis's work for several years now. He really has a lot of creative ways of looking at kind of non-traditional data and methods that we have for understanding protein function and structure to help us interpret missense variants, which, as we all know, obviously is a major challenge in the field. Today, I think he's going to be talking about a couple of different vignettes on various techniques for doing that. And so, Dennis, I'll turn it over to you.
Yeah, thank you very much, Jason, and the whole team for inviting me today. Let me see. So, could someone please confirm that you can see my screen?
Yes, we all see. Yeah.
Oh, perfect. Okay. So, yeah, I give a talk today about this primary focus on bioinformatic methods for interpretation of missense variants. And we do also other research, but today is really focused on that. And the overarching goal of these methods is that they are transparent, that you can, as a biologist or medical doctor, get some intuition why these methods say that these variants look more likely to be of importance for function than others. And yeah, I will not discuss only the paper which was advertised. I will present 2 additional studies because it's a little bit chronological work. So, so the first study, which was also the one which is advertised and just recently came out, is actually very old. It's like 3 years old, and was even done when I was a scientist at the Broad Institute. And probably the last study I did did a lot of things myself. And then I present the follow-up studies, where the second one was actually also recently published, which went way faster. And then I show some new things we do. Okay, so the first project basically uses gene families for the first time as a unit to make sense out of missense variants. And it all started a couple of years ago. So by training, I'm a geneticist focusing on epilepsies and neurodevelopmental disorders. And we try to figure out which genes play a role in these disorders, and how do variants in these genes lead to the molecular pathology causing disease. So one thing which we noted is already like a couple of years ago is that many of these genes sound similar. And this is not random. This is because many of these genes associated with epilepsy, for example, are ion channels, for example. And genes which have similar function have frequently also similar nomenclature-based how we as geneticists call these genes. And then you can really see that there are many related proteins. So throughout my talk, I will discuss the concept of gene families, and that's why it's important to define that here at the beginning. So I will talk about genes which are related to each other because they share evolutionary-wise a common ancestral gene. And I would call these genes which are part of a gene family paralogs. And throughout my talk, we focus on within human genome paralogs, so only related genes which belong to the same gene family in humans. So why paralogs? So that's one thing which is, well, was until recently actually not appreciated at all, is that if we look at you know, conservation in terms of variant prediction or just assign and function through evolution, we usually look across species, and there were also some let's say bioinformatics scores which also incorporate paralog information, but no one actually has really did a one-to-one comparison. What is the difference between conservation, let's say, for the same gene across here these species, or this gene to their gene family members, so the other paralogs? You know, how different is the conservation spectrum if you perform, for example, a linear protein sequence alignment? And this difference is tremendous, and this is actually not really appreciated. So if you look, for example, at this voltage-gated sodium channel gene SCN1A, and you compare the linear sequence alignment, for example, here's the first amino acid, here's the last one, and you can always see the 3 species. You can see that the conservation on amino acid sequence level, these 3 genes are almost perfectly conserved. Whereas if you perform within humans an alignment from SCN1A to SCN11A, so we have 10 of these voltage-gated sodium channels, when you perform this alignment, you see here now that there are many gaps and you have hardly any conservation. So there's a very different, you know, kind of conservation going on. You see like some sites which are super conserved, like here, whereas, you know, also like large fractions are conserved here, but here not at all. So, and this is now interesting because when we look now at population references and look where do we find variants in the general population, we see that first of all, we see a lot of variants in the general population. So on the other hand, if we look at disease databases, we know that many of those sodium channel genes are associated with really devastating disorders. So logically, it cannot be that everything which is conserved here across species is of importance in humans. So which this observation like a couple of years ago made us, you know, build the hypothesis that maybe not so much sites which are, you know, that maybe the parallel conservation gives more information in terms of distinguishing between essential sites for function in humans compared to, for example, autolog conservation, which, you know, in many genes, a lot of things are conserved. So we started this project coming from a disease angle, because if we could refine the essential sites in a gene, then we might be more powered to do gene discovery. And so we originally started with 10,000 data from 10,000 trios, where we have 2 unaffected parents and affected child. And we have the list of de novo variants, which are present in the unaffected children. So well, we collected data basically from these 10,000 trios, where the index case, so the child would have either developmental delay, autism, or epilepsy. And just for those who are not working on brain disorders, these disorders have a very similar spectrum of genes associated with these disorders. So, and traditionally, how we in genetics discover now new genes which have a de novo variant burden, we use this approach developed by Kathleen Samocha and Mark Daly et al. A couple of years back, where we used empirical data and the organization of the genes in terms of nucleotide context to, you know, to generate an estimate how many de novo variants you would expect if you sequence a certain number of trios in the offspring. You know, how many de novo variants you would expect in the offspring if you sequence a certain number of trios. And this model has been benchmarked quite a lot, and is basically the foundation of how we identify genes today in monogenic pediatric disorders in particular. So, for example, if this model says you would expect in 100 trios in a specific gene 1 variant, and you observe here in this, like in this example, 6, you can do a Poisson test and see if this is statistically unusual. And if it would be unusual, you would conclude this is because these individuals which we look at are not individuals from the general population. These are children with disease, and that's very likely to be a disease gene. This is the underlying principle, and this has been benchmarked also against trios where the child doesn't have a pediatric severe disorder and it doesn't give any new insights. However, one thing what we noticed recently is, although we call these disorders only neurodevelopmental disorders, autism, epilepsy, and so on, all these terms are umbrella terms for many more disorders, probably in the hundreds of thousands. So what the challenge is, even if you look at 10,000 trios, there's a good chance that even truly disease-associated genes will have few variants. So, and our hypothesis was maybe we can build this single gene burden test to a whole gene family level burden test. And that's very similar to this one. You basically sum up the expectations for individual genes, which here in this case are genes which belong to the same gene family. You would have which gives you an expected number of de novo variants in 100 trios, for example. And then you look what you observe, and if there is true signal, so that multiple members of the gene family contribute a little bit, you might get a significant p-value and could identify or a gene family with significant burden, which might imply new genes or give at least good candidate genes. And the beauty about this project is that we can go even one step further, because what I mentioned before is, in this simple example, is that for evolutionary conservation here, for example, measured by the GERB score, we already know that they are amino acids are very well conserved. But we already observed before, if you look at cross-paralogs, that there are many more amino acids which are not conserved. And we tested this now across all genes which belong to a gene family. We first looked only at their autolog conservation, so across species. And we see, we look at around about 9,500 genes here, that most of these amino acids are relatively strong conserved. However, if we look at these 9,000 genes and generate a paralog conservation score, we see that only half of the amino acids are conserved. So what we saw in these individual examples is also true on larger scale. So this is not beautiful, because we could generate a score which scores every amino acid in every gene belonging to a gene family. And with this, we can not only look at the gene family burden test, and in addition to that, we can just separate things. We can separate every amino acid in this gene family and gene into those which are parallel conserved, so identical in every member in the gene family or very similar, to those which are very specific and so not conserved for a member in the gene family. So because we have these alignment-based scores, so which we generated for 2,800 gene families. So, and with this, we can ask now for the first time a very simple question. Are our disease-associated de novo variants do they hit parallel-conserved sites, or do they hit not conserved sites? And this has not been done before. So if we now perform like a gene-level burden analysis and test 2,800 gene families for de novo variant burden in these 10,000 trios, so we see that we do not find a single gene family, here's the multiple testing cutoff, which is significant for the not conserved amino acids. So however, if we look at gene family, so parallel conserved amino acids, we find many gene families which show exome or 2,800 gene family tests wide significant burden. So if we now look at this, so in total we find 43 significantly enriched gene families, of which which individually in these we would have expected only 50 de novo variants, but we found almost 800, which is a 15-fold enrichment. So luckily, we were also available to find an additional 2,000 trios where the index child doesn't have a neurodevelopmental disorder. So basically as a control. So in these 2,000 trios, we see 10, we would expect 10 de novo variants, but we only observe 5. So, so we would expect 10, but we only observe 5. So this is not significant. So many of you might now say, yeah, okay, that's cool that you can show that gene family, the variants are at parallel conserved sites. It's interesting, but how meaningful is that for new disease gene discovery? Because these 43 gene families might contain many already known, disease genes. So if we now remove basically from our 43 gene families all genes which have been previously shown statistically in an exome-wide approach that these are individually significant genes, exome-wide, in a de novo trio statistical test, we still see a five-fold enrichment, which is highly significant. So there seems to be in these gene families additional risk, which we could, unravel with this approach. And here's now a list of these genes. So the study is a little bit older. So many of them have now been shown also in individual studies that they are relevant to the disease. And here are also now these shown, which are also, brain expressed and deleted for, or have variant constraint. So, and the good thing about these paralog conservation scores, which we generated from these 2,800 gene family alignments, is that we have now an amino acid position score. So, which is relative and which is very easy to interpret. So, what you can see here now is this line, which is showing that basically is the median value about the Z-score. So you can see now here amino acids which have a very high score for this potassium gene family and casein Q2 gene. And you can see basically in this gene casein Q2, all amino acids which are identical in other casein Qs, like casein Q1, casein Q5, so other potassium channels, are fully conserved. And you can nicely see that this aligns with the known domain of, for example, this ion transport domain. However, what we also can see here is that not every UniProt information, like domain information, for example, is 100% correlated. So we felt that maybe combining this information with the domain information with paralog information might give some additional resolution. And it's very easy to interpret because this this line of amino acids seems to be is conserved in related protein mass. So thinking from an evolutionary perspective, has been protected through millions of years is important. Okay, and here nicely what we see is in this study that we have patient de novo variants in these paralog conserved sites, and for example, synonymous variants don't follow that. So here are a couple other examples how we can now visualize the variants and can help maybe to generate some hypotheses, why in particular certain regions are more paralog conserved than others. So in this study, again, I want to summarize that we present the first time where someone used the gene family as a burden test and could show that paralog conserved sites are the ones which are more implicated in terms of de novo variants and likely function. And we could, prioritize some genes and candidate genes. So, and that was recently published. And so now I'll move on to the next study. So basically, we make this observation that we see that there are some sites which are paralog conserved. And we thought maybe we can combine this also with an additional line of evidence for that something is important in a region of a gene, and which is the distribution or like basically the clustering of patient variants and the absence of control variants. So the following study is basically the next logical step after we develop this paralog conservation score. So again, here's a reminder, we talk about gene family, which means related genes in the human genome, and we call them an individual gene, we call it paralog because they are related. So in the next approach, as I just mentioned, we look at the paralog conservation, but in addition to that, we now include also information from the general population. So here we are basically checking if there are regions which are enriched for patient variants, but depleted for population variants across the whole gene family alignment. And this is what I mean with this. So before in the previous study, we generated this alignment of this gene family here. For example, this gene family of 10 amino acids, 10 genes, so 10 paralogs. And before we would score if this amino acid is, for example, fully conserved and it's very high, whereas this would get a low score because it's not really conserved. So there's like this amino acid is missing in some of the members. So, but here now we are doing a different approach. So what we did is we took variants from Nomad and look now. So Nomad is the largest database for human variation at the moment when it comes to rare variation, from 140,000 individuals, where we have data variants or missense variants for every individual sequence. And we combine it with these two largest patient databases, so ClinVar and HGMD. So what we do now is we have this 2,800 gene family alignments, and now we just ask every time in this alignment if this individual amino acid, again, like here, you have the amino acid sequence, if this amino acid is mutated in the general population or not. If it's mutated, he gets a 1, for example, just in this O. So in this example already, you can see that there's a region which doesn't have variants in the general population, which is already, pointing us in terms of data that this could be important. That's why healthy people don't have variants here. So in the second approach, then we did totally the same thing, but we did and map now, but this time we mapped patient variants. And what we would expect if some residue is very important across the gene family, we would see many patient variants in that region. So, and then we simply performed, we basically performed the burden analysis. And what we are looking for are regions which are low in variants from the general population across similar residues in the gene family compared to patient variants. And with this, we can do a statistical test to identify regions which are enriched for pathogenic variants. So, so here's now the setup. So we had from our previous studies 2,800 gene families, and did a parallel also an analysis only on gene level, just to see how strong the gene family approach performs over and above single gene analysis. Then we have these alignments. Then we set up everything into a web tool, a Shiny tool, which is based on R, and mapped in 2 million Nomad variants in these alignments. And we mapped in around about 100,000 patient variants. And hopefully we could identify regions which are significantly enriched, and then we get a p-value for the region even. So that you are able to see, how important a certain region is. So this is how it looks. So what you get here at the is that you have the first amino acid here, and here you have the last amino acid, and you have this alignment of these, for example, here the sodium channels again, of these 10 amino acids, of these 10 proteins. And you have to imagine that this would be the index alignment position. So you basically you have your positions here like Asimone, you have your genes here, Asimone 2 or 3, and so on. So like this. And then you have here the first aligned amino acid, and goes until to 2,500 to the last amino acid. And then you can ask for all of these genes, how many variants in the general population do you have here shown in this green bluish color, and how many variants you have for ClinVar and HGMD, so patient variants. So and so all these databases are not perfect. So you would hardly expect that there's a region where you have never a population variant or never a patient variant. However, what you're looking for is really anti-correlation. You want to look for something where the population variant goes really down, and we have a peak in patient variants across the gene family alignment across all these guys. So and if this is happening, then we get a very significant enrichment. And this enrichment is shown here for on this Y scale. So shown here in the red bar, and here's the p-value enrichment. And so with this, we can now identify regions, and if a region, because we run here sliding windows, and if this region is statistically significant enriched after multiple testing, so basically accounting for all the sliding windows we do per gene family, so then we call it a PER, so a pathogenic variant enriched region. So, and as you can identify, see here, we identify a bunch of regions in these in this gene family. So, so again, when we look, so basically we're looking for anti-correlation of patient versus control variant across the whole alignment. And this is how it looks like a little bit in more detail. So we can, on the web page, you can also scroll through it and see it a little bit more in detail. So you have an alignment position of the whole gene family. And for example, at position 457, we have a leucine, which is, and this alignment position is representing position 417 in SN1A, which is position 419 at SN2A, and so on. And we see, for example, that at this alignment position, all the time, this amino acid is conserved. And this is per position 2. And interestingly, no patient variant has been reported here yet. However, in the neighboring regions for this amino acid, you can see that's also fully conserved. However, here we have already patients reported with, for example, like pain disorders, muscle disorders, heart disorders. Yeah. And for example, because the sodium channels, basically every sodium channel is associated with at least one gene, with at least one disorder. So many of them are associated with multiple disorders. And you can see, for example, that here is a patient with a variant in SCN8A at position 407. And we have a patient with a long QT syndrome, so heart disease, at position 409 SCN5A. So which gives you also some, if you want to, get more, give a feeling if this variant is important, you can basically get more confidence because you know that at the same analogous position in a related gene, also patients have been reported, which is quite nice. And I don't talk about this today, but you can basically look even at the functional outputs. For example, that variant could be a gain of function variant of the ion channel. And what we learned in preliminary data at the moment is that this variant is also, this variant in this gene is also very likely to be a gain of function. So it seems across paralook, even the pathomechanism on biophysical level is conserved because the structure is so similar. And this allows even to develop a machine learning algorithm which uses functional data from one paralook and predicts with like 80 to 90% accuracy the functional output in a related protein, but it's not part of this talk today. It's pretty cool. Okay, so and the cool thing is also that since we do a sliding window, we can identify now regions and can even prioritize amino acids which are within a pathogenic variant enriched region where no patients have been reported yet. And we think because of the statistical enrichment and the conservation that this is a very important region and can help their interpretation. So just to brief go to the statistics we have in the paper, you can see, so we identified more regions like using a gene family approach compared to an individual gene burden test. If you just do the sliding windows for a single gene. So we only identify hits in 250 genes, whereas we have 465 in gene family approach. And we capture also way more residues which are within a pathogenic variant enriched region. So, and this enrichment has been done, I told you, with ClinVar variants. However, if we use a newer version of ClinVar, which didn't, where variants were not part of, based on the algorithm development, and we look now which variants have been, in this disease variants, have been classified as benign or pathogenic, we see a 106-fold enrichment for that a variant is classified as pathogenic compared to benign if we look in this independent data set. So which gives us also some confidence that the regions we identify are useful. So, and the website is available. You can access it. You can type in your gene of interest. Then you get these alignments and you can see these pairs for the whole gene family, but you can also select an individual gene to identify those. You can also, zoom into a region and then explore, where which amino acids from each member are reflected by this, which diseases are known for any of the family members at this alignment index position. And finally, then can go even further. And since we score look at every per individually, we give them numbers, and you can look, which genes contributed to each per, and which disorders are contributing to those. So giving you some more transparency And that has also been recently published in Genome Research. So finally, I just have to check my time. Yeah, 50 minutes. So okay, there's a summary. So before we showed that parallel conserved regions are enriched for variants, but now we look even that we can show that if we use these gene families alignments and map in variants from patients and controls, we can identify very powerful some regions in the in these genes and gene families which are clearly enriched for pathogenic variants, because the distribution of patient variants is very similar along the linear protein sequence for related proteins, so genes which in this case are part of a gene family. And same is the same anti-correlation is observed, or similar anti-correlation is observed for nomad variants, so population variants across the alignment. And it's quite interesting. I think we can now quite compelling show that really the parallel conserved sites are the ones which are very important for severe disorders. And we did an analysis where we looked, and I don't show it today, at across like dominant diseases, recessive diseases, and different disease organ systems affected by different types of disorders. Maybe these are just subset claimed by ICD codes, for example. And we see constantly across all diseases, even late onset diseases, that the disease-associated missense variants are within the parallel conserved regions. I would never argue that there are exceptions, but overall, statistically, it looks at really the parallel conserved regions, and makes somehow sense if you think about that you have a gene family of many members, and you have some sites which are still, since millions of years, although they are already, you have this gene family in multiple proteins, but still in humans, they have sites which are not which are identical, which means throughout evolution, individuals who had mutations at these sites were kicked out of the gene pool and could not reproduce. So there's a lot of protection going on, protecting these, I guess, infrastructure sites for these type of proteins. Okay, finally, I will use the last 10 minutes to show some preliminary data, which has not been published, and I would ask the people online to treat this a little bit more private. So, as I mentioned, we are a genetics group focusing on focusing on brain disorders, and with an emphasis on pediatric disorders. And we noticed in the recent years that neurodevelopmental disorders, although they are individually rare and they are relatively common, and the beauty about genetics and on these kind of disorders is that we have identified many genes in the last 10 years. Actually, several have been identified before, but due to the new sequencing technologies in the last decade, we could identify most of these genes in the last 10 years, which is cool, right? So, but the problem is we identified now that we, identified all these genes, and now people want to clinically test them. But the thing is, since we identified them just recently, we hardly know how they work. And that leads to the problem that we test them clinically, but we don't really know what are the essential sites in these genes. So, and one thing is, I mentioned this before, is that many genes which have total different biological function, and besides that they are expressed in the brain, but at the end, how they have different roles in the cell, they can lead to a very clinically similar looking disorder. And even on the other hand, if you look at some genes, we have variants in the same gene, but the disease is very different. So there's also heterogeneity on gene level. So, and okay, that's what I've just mentioned. And so, and as in addition to that, in literally every gene in the human body, regardless how disease associated it is, we always have variants which do not cause disease. So there's a lot of things to understand in terms of function. And so what makes a variant to causing disease, and what leads to gain of function, and what leads to loss of function, because these kind of information are really important when it comes to therapy. So, or predicting the outcome of a patient. So many bioinformatic approaches focusing on, and when it comes to genetic variants, focusing on pathogenicity, which is amazing what happened in the last years, because these variant classification scores or prediction scores are becoming really good. So they really approach clinical utility. However, when it comes to the next step, which is building, as a biologist, hypotheses to study the new protein, or as a clinician, getting eyeballing a variant or understanding the rationale to become more confident behind pathogenicity classification, many scores are not really useful because, or don't go one step further, because you have the score, but you don't understand why this score is giving the score. So, and we thought that's something we can work on. So again, just to remind you about the situation. So we have identified around about 260 neurodevelopmental disorders, 30 genes which are monogenic. However, as you can see here, most of them have just been identified. The vast majority have just been identified in the last 10, 15 years, right? So, and that translates to the following problem. For these new these disorder genes, there are hardly research studies out. You can see the largest category of genes has, which is more than 100 of these neurodevelopmental disorder genes, less than 10 research publications have been published, which is, delays translation into drugs or, more confidence variant interpretation tremendously. And so our goal was, to develop something which could guide biologists and clinicians in prioritizing regions in the biological active molecule of the gene, which is a protein. So we thought maybe we can do something large scale, which points to as an essential region in the protein, which can help facilitate molecular research for drug development or variant interpretation. So I mentioned this already. So the classical ways how people in bioinformatics research prioritize regions for function in the general on proteins are basically three. If you just want to, you look at conservation across species. You look now, hopefully, at conservation across paralogs, and you look if there is a depletion of variants in the general population. And we thought, hey, that's pretty straightforward. We can use our parallel conservation score, which we mentioned here before. And we can also use a regional depletion score, which identifies regions like this in the linear protein sequence, where we have hardly variants in the general population, like it's called the MTR score. We can use these two scores and look for where they have overlap. So evolutionary conservation, population conservation. And so we have our PARO-Z score, we have the MTR score. And we thought, hey, maybe we can go a step further, and we can find for all neurodevelopmental disorder genes where possible, protein structures, and we can perform a spatial enrichment statistic, or where we can identify regions which are enriched for patient variants compared to control variants. So then we have like some kind of clustering, which points us that this region might be of importance. And then we have this population information and this evolutionary conservation score. So, and we thought if we have, multiple lines of evidence from independent sources of, let's say, pointing towards importance of a certain region, we call this an important region, and that could be useful. So first of all, we started by collecting neurodevelopmental disorder genes, and we focused on published and recently bioarchive deposited studies, which is covered through a de novo approach, 246 high confidence exome-wide significant neurodevelopmental disorder genes. Probably there would be also more genes, but that's the way we did it to be comprehensive. So, and then we selected, because we wanted to really close to the, biological active molecule where we can infer function from, namely the protein, only genes where we have at least 50% coverage of a protein structure. And, you see that's already like a big, filter step, because from these 246 important neurodevelopmental disorder genes, we have only 56 protein structure, which covers at least 50% of the protein. So we needed to do some extra work to fill a little bit the gaps. So what we did is we collected already homology models for which other people have generated and did some filtering for quality to increase the number of proteins. And additionally, we predicted ourselves the modeled structures where we could for an additional set. So in total, we ended up with 163 neurodevelopmental disorder associated proteins where we have at least one, sometimes multiple types of protein structures. Then we took the evolutionary scores and the population scores, and in parallel, we generated a clustering score and combined all this information. So we took the protein structures and mapped the evolutionary conservation score, the paleoconservation score, and the clustering regional score to identify, and then look where all these scores are overlapping to consider this as a very essential region. Just here as an example, we have here the paleoconservation score in the linear sequence space. Then we have the MTR score in the linear sequence space, and here you can see population constrained region. Here you can see conserved regions. Here we see the clustering in the structure, and then we look where all these kind of analysis overlap, and then we call it hotspot 3D. Probably we have to change the name because something different will have. Also, protein structures has just been published with this name, so we will change that. So, but anyways, so we're looking really for the most obvious important thing in each protein. So here's a little bit statistics. So if we now perform the linear-based scores, if we transform them into 3D and normalize them, we can basically see that the more paleoconserved, this is diagonal, each region is, the more variants we see enriched for patient variants over control variants. And here's this, if we look at only single proteins, here if we do predicted structures, the homology models, and here's at predicted structures, and here's the whole complexes. And we see a similar picture if we look at population constraint. If we transform this into 3D, we see always these gradients to some degree. Okay, that's nice. So what we did on top of this is now we took individual de novo variants, and this is important to note because here's a comparison for the enrichment of de novo variants over population variants, or at least not variants enriched for brain disorders. And this is, not all of these variants will be pathogenic. That's why our fault enrichment will not be so high. So, but what we can see here is now that our hotspot regions and all these kind of protein structures, again, single protein structures, model, predicted structure, and complexes, we see always in like a 6-fold enrichment for de novo patient variants over, the other type of variant. And that's pretty nice because not all these variants are pathogenic. And so if we have a de novo variant here, that seems to be really of importance. And I don't have this here today, but we recently did also like the classical ROC curves and so on. And it looks really, if we add our hotspot 3D to existing scores, the models improve, and that's really nice. So now an interesting observation is, we talked about function, and if we talk about function in proteins, an easy way to, which is not comprehensive, but to get an idea what are functional important regions in the protein is look at uniprot features. So like information about amino acids, what they do in a protein. And strikingly, what we see is that, and in the single structure, in all kinds of structures we looked at least 50% of the protein is has a uniprot feature. And so meaning for us, that make very clear that this uniprot, having a uniprot feature associated with an amino acid is not really helpful because almost half of the protein has this. And we already know that only a subset of uniprot features are really of importance. And so if you, for example, look at our hotspot features, we see that 6% of the protein, for example, of a single protein has our hotspot 3D score, and the vast majority has also a uniprot feature. There's a strong enrichment. So like 90% or so, compared to 50% across the whole protein. So our hotspot is enriched really for UniProt features. But there's also some amino acids where we don't have a UniProt annotation, but it's within our hotspot region. So this is really for molecular biology, I think, biologists region to take a look at and discover new insights about these proteins. So anyways, so it's really important to know, which UniProt feature is really meaningful. And in our variant set, we can simply test that. And we see among neurodevelopmental disorder-associated genes, particularly the UniProt feature transmembrane seems to be most strongly enriched. And this is particularly reflecting these kind of ion channel genes. Makes sense. So, but we also see, like, for example, that metal binding sites are really significantly enriched in our data set. And this is reflecting these phosphatases. So, and so on. And some other regions are just depleted. And this is, a very stronger enrichment compared to any kind of UniProt feature, which is an enrichment of 2. I think we don't have that here. No. Okay. So, but it's important to note that this is really data set specific. And if you would look at different types of genes, you would probably see a different kind of enrichment. But it was quite nice to dissect this here a little bit. So finally, this is now where we feel this is really useful. We can generate now these kind of visuals for 163 proteins. Most of them have not been studied in protein structure space. And you can see nicely here, in red, where, for example, we have the complex here, and you can see here where all the patient variants are located. And in purple, you see here the hotspot 3D, and like here as well. So it's so we have now basically a map where we think where the most important region, statistically speaking, from multiple lines of evidence, is for more than 70 genes, many of them not studied at all. So that hopefully can guide biologists and generating some hypotheses like this one. If we look at this GNAU gene B1 complex, we see that there are multiple proteins involved, including HTBR1 and these 2 proteins. And we can see that here are our patient variants, and here on blue are our control variants. And if we do our hotspot score, which shows us then at the end that apparently our evolution and constraint and so on scores show that these amino acids seem to be the most important ones, which are quite interesting because this region is, for example, interacting with a GTP. So here we can model in the GTP binding sites based on the structure for mouse and understand now, so but just with simple protein structure biology, we can, test the hypothesis, that this region is, which is, this is a GTP binding, GTP type of protein. And you can see really that here, the GTP is binding. So it nicely explains why this looks important. And you really can see that the amino acid face this molecule. So, and the other region is because is potentially because there is a hydrogen bond. The hydrogen bonds are also statistically speaking, are very important, interacting between the alpha and the beta part of the protein. So you can see this here, this interaction. So the whole point of this is, this annotation which we develop like this allows now biologists to develop hypotheses in an environment which supports statistics. For example, this is a glucose transporter, and I just want to go quickly explain this. And this is very an important gene because if you give the child ketogenic diet, it will be basically fine as long as the child eats ketogenic diet. But on the other hand, if it doesn't, the brain doesn't get sugar, and the child will develop a really severe developmental delay. And if this goes on for a couple of years, it's irreversible. So,
Dennis, just a quick time check, to leave at least 10 minutes. We at 51
okay,
right
okay. Now. Okay. So just show at the end, here's the sugar binding site, which you can model in, and you understand now that the inner tube is important, and we can, which is like a hotspot region for us, and it makes sense because it's the molecule interacting here, and there's also another molecule interacting at this site. And we can look at other complexes and identify also, the region, and can identify that this is a metal binding interaction site, and so on. So just in summary, we develop now this kind of infrastructure which can develop hypotheses for many of those genes. Okay, with that, I'm done, and thank the organizers for letting me speak, and if there are questions, happy to address them.
Yeah, that's great. Yeah.
Great. So does anybody have any questions they'd like to ask Dennis?
Should I stop sharing?
Yeah, you can stop sharing the screen so this way we can see you when you. It'll be great. I guess one question from my end. You mentioned that transmembrane domains basically as number one on your list, right? In those hotspots, right? I mean, the transmembrane kind of broadly covers a lot of scenarios, right? I mean, they could be right in the channel, could be structural to the transmembrane domain itself, could be exposed to the fatty acids in the membrane, right? Do you have a better sense where they cluster?
Yeah, for in this set, since many of these brain disorder genes are ion channels on transporters, it's basically the pore region which is facing inwards. So, but this seems to be true for many of the ion channel genes, but some of them have like some molecule, some motifs which are functional, and there it's not only the pore.
How well those classifications in general work? I mean, there's a lot of more recently, crystallized or by cryo-EM done complexes, right? When we learn quite a bit about things on the interface, et cetera. When you look to the databases, do you think the classification really stretches to where the current knowledge is?
I mean, every knowledge which is more like publication level, where people have investigated certain protein regions and mapped them out nicely, or will be better than these large scale annotations. But I think in the absence of high quality individual level kind of annotations, the UniProt annotations are good, but there's room for improvement for sure.
We just look at final thing. And how do you guys follow up with validation? So do you engage in collaborations, or do you just kind of leave it out for people to grab?
Yeah, it depends on the project a little bit. So one thing is that for the epilepsy genes, which is probably the disorder I study the most, we are part of a multi-center project grant which is basically functional testing, where we combine the functional readouts with the protein features and try to make sense, and also develop composite scores. So we do functional testing. But the interesting thing is that functional testing turns out, when you start to do a lot of benign variants or normal variants, if you test them functionally, at least in ion channels, suddenly they also become functional. So it's really hard to distinguish the cutoff when is a functional equals pathogenic, and when functional is tolerable. So even for monogenic disorders.
Right. Good.
I have a question, Dennis. I'm curious about the paralog conserved regions. So I think I'm trying to think of the biological model, because on the one hand, you could imagine that the more conserved the region is, the more relevant it is to disease. But then you could also hypothesize that you actually are interested in the divergent regions, right? Because within a human, like cell type specificity and things like that are often relevant to disease. And so it's not obvious to me that the conserved regions would be where you'd expect to see the pathogenic variants. And I guess I'm curious as your take on, do you think this is something that's general for rare diseases as well as maybe more common? Do you think it's specific to rare neurodevelopmental disorders and severe things, or what your take on that is?
Yeah, so unfortunately we never tested this, but my personal opinion on this is for really these disorders where one variant kicks you out of the gene pool really under selection, it's this infrastructure sites and this diversity which you still have comes from the tissues or cell type specific expression. So many of these genes are really specific. Even if you have 10 members, some of them are really in one brain cell type, although you have 4 in the brain, but some are really in the brain and different. And I think when it comes to more milder variants, or to the question, if you develop disease X with let's say like epilepsy with 50 times seizures a day, or if you develop a disease with 3 times seizures a day, that can vary, on the degree of conservation. I could imagine that. And I think everything which is more, a population common disease, the parallel conservation becomes weaker. So because here this can be tolerated, evolution is not working so strongly on that.
Right.
And especially late onset disorders. Although we see some signal, but it's definitely weaker. The more early the congenital disorders, the stronger this observation is we have.
I see. Yeah, I mean, because I saw, I think there was a talk in our division about how when they were looking for diseases and, genes that might be involved in like autism, they were looking specifically for regions of the genomes that were only conserved in the later primates, whereas obviously the super conserved you would, right? And I'm do you think have you guys explored things of more fine-grained types of alignments where like rather than looking at the whole family, like you look at conditional conservation within those that are brain or anything like that?
Yeah, this is something what we're doing at the moment. So we generate on the one hand mega gene families which are way bigger. And on the other hand, we do also cross species. You can, for example, take a gene family of mouse, gene family of humans, and so. And on the other hand, we also do more tissue-specific gene families, because that allows us to, at the end, then do better statistical burden tests. If there would be signal in ion channels and and there's a brain disorder, that signal should be in the in the genes which are also expressed in the brain. Everything else would just be noise. We're looking at this also. We are trying to improve the missense variants, which we select, for example, with this PER approach, because some will be just very similar to a synonymous variant because the amino acid exchange is from a positive to a positive, and that might be tolerated. Whereas if it's more, detrimental, that should not be seen in the. So we're tweaking in all kinds of angles. That's definitely not final what we just presented today.
All right. Anybody else have any other questions?
Yes, one question. So Dennis, it's really like an interesting talk. So I have one question. So when you actually calculate the parallel conservation score, so do you consider the family size? Because from my understanding, so for the gene family, if you most of the genes actually from the same gene family have similar function, so they actually have the complementary function when you knock out one gene. So I was thinking maybe gene families which have no gene family or have very less gene family members, then those families maybe are more important than the other families.
Yeah, so that's a very good point. And so what we do is we take just gene families from the HGNC defined on ensemble-based defined gene families, and then to get good alignments, we do a cutoff that the sequence have to be at least 80% similar. So that's what we do. And sometimes we didn't do a correlation between constraint and gene family size. That would make sense. But what others have already shown is that there is some kind of parallel compensation mechanism for some gene families, and that those genes which are not part of a gene family, that they are super intolerant for stop codons, because there's no one doing the job if that one is not there. So we didn't really incorporate it, this information in the alignments we have yet, and did not test it. But others have shown if it's not part of a gene family because it's a very specific protein and you have a stop codon in those, that's more likely to be a disease-causing variant or gene.
Thank you.
Great. All right, I think we're at time. So Peter, any last words or announcements?
I mean, not for me. I mean, we have another seminar 3 weeks from now at the same time. If there is more, we're going to announce. And again, go to RC Wiki where everything is recorded. I've showed before people started. So get it on your iPads, iPhones, and you can listen. Well, we're not going anywhere nowadays. So I don't know when, but I'm sure you guys can figure it out. If any last minute questions, just feel free right now to unmute. Last opportunity. Otherwise, we conclude, and thank you, Dennis. Thank you, Jason, for joining us today.
Yeah, thank you very much, Dennis. It was a great talk. All right.
All right, thanks.
Take care, everybody.
Bye-bye.
Bye.
Bye.